All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members who are attending today. Your ASPB member dues help support making this webinar possible. For any of you who have not yet joined ASPB, you can join today and use the discount code %10 and to receive a 10% discount on your membership registration. ASPB members get access to many great benefits. And so definitely learn more and think about joining ASPB today. And if you're not already. You can learn more about ASPB and all the many great opportunities we provide at aspb.org. Next slide, please. As you know, and for many of you who have been joining Plantae webinars for quite some time, this webinar is recorded, and so you can watch them on our Plantae YouTube channel. And so want to make sure that you check those out, especially if you're looking for many additional great content. Before we get started, I'd like to highlight a few housekeeping items to make sure that you get the most out of attending today's webinar. If you are experiencing technical difficulties, please let us know about those by emailing me at jpadilla@aspb.org. If you have questions to our guest speakers, as I'm sure you're going to have them after hearing them talk, or maybe you already came with great questions, please use the Zoom Q&A section and type them. Our moderators will be sharing them with our presenters, and we'll be going over them during the Q&A portion of this webinar. If you are having trouble connecting or need to leave the webinar early, as I mentioned, this webinar is recorded and will be made available on our Plantae YouTube channel. So again, be sure to follow our YouTube channel to receive content notification when content is posted. And without further ado, I'm going to now turn over the floor to our wonderful Plantae Fellows who have spent great energy organizing this really informative webinar. And without further ado, take it away, Plantae Fellows.
Good morning, everyone. Welcome to our webinar today on the world policies in plant science. And this morning we have an exciting webinar planned for you. And we have three speakers, Dr. Susana Valderrama, Professor Casey Bansal, and Dr. Felix Moranto Barrios. I am Jillian Rowe, and I'm joining from Kingston, Jamaica. I'm a 2024 Plantae Fellow, and I'm also a doctoral researcher at the University of the West Indies, St. Augustine in Trinidad. However, I'm based in Kingston, Jamaica. So just a little bit about our webinar for this morning. We will be having, the speakers will be presenting on their topics. And previous slide, please. And if you have questions, we will be taking your questions at the end of the presentations. Feel free to type your questions in the Q&A box, and then we'll have the conclusion. So we look forward to hearing the presentations of the three speakers and then answering your questions at the end of all of the presentations. So thank you for that. Next slide, please. So as a doctoral researcher, my research is focused on the soil microbiome and plant microbiome of ginger in Jamaica and developing a novel biocontrol agent for the control of the ginger rhizome rot. Next slide, please. If you're interested in world policies in plants and you want to find out more information about these policies and get involved, you can contact the ASPB Science Policy Committee. And they are responsible for assisting societies as well as other researchers, policymakers, developing strategies and objectives for support of basic plant science and coordinated efforts with other societies, such as the Global Plant Council. And if you want to get more information, you can contact any of the chairs of the ASPB Science Policy Committee. Next slide, please. We compiled a list of some of the world policies in plant science that we found were relevant to plant science researchers and plant biologists. And some of these include the Nagoya Protocol, the Convention on Biological Diversity, the Convention on International Trade in Endangered Species of Wild Fauna and Flora sites. Others that are related to plant protection and health, next slide please, include the International Plant Protection Convention, IPPC, the Cartagena Protocol on Biosafety to the Convention on Biological Diversity, and the International Treaty on Plant Genetic Resources for Food and Agriculture, and the SPS Agreement. If you have more world policies in plant science, please remember to reach out to us. Feel free to reach out to us, and so we can put together this list of world policies which have become relevant to your research. Without further ado, I'd like to introduce this morning our first speaker, Dr. Susana Pereira Valderrama, who is the Program Management Officer at the Cartagena Convention Secretariat of the Caribbean Environment Program, which is based in Kingston, Jamaica. Dr. Pereira Valderrama is the Program Management Officer at the moment, but for the past few decades, she has been involved in monitoring programs for the marine protected areas and also for monitoring ecosystems and design projects. So without further ado, I'll welcome Dr. Pereira Valderrama to speak to us this morning.
Good morning, colleagues. Can you see my slides?
Perfectly. Thank you, Susana.
Yes, we can. Please go ahead.
Yes.
Okay. Well, good morning or good afternoon, and greetings from the Cartagena Convention Secretariat here in Kingston, Jamaica. I'm very happy to be here with you today, and I want to thank colleagues from the American Society of Plant Biologists for inviting me to this session. In this presentation, I will provide some information about the Cartagena Convention and in particular about the Specially Protected Areas and Wildlife Protocol and its objectives. And I also will present the articles of the protocol that address the conservation and sustainable use of flora and fauna species. And I will also share some our perspective on how plant scientists can incorporate the protocol into their research and explore opportunities for collaboration. So in 1974, the United Nations Environment Program launched the Regional Seas Program to address pressing environmental challenges facing for our oceans and coastlines. And this initiative became the primary regional mechanism dedicated to conserving marine and coastal environments, and is one of the oldest UNEP programs. This year marks its 50th anniversary. So as you can see, there are regional assist programs all over the world. And this UNEP program was created to address marine pollution and the degradation of shared ocean and coastal areas through targeted regional actions. And this program emphasizes the need for environmental strategies that reflect each region unique characteristics. So this initiative regulated and fostered collaboration among governments, scientists, civil societies, local communities, and indigenous people. So of this 18 regional assist program, the United Nations Environment Program administered 7, and the Caribbean Environment Program, also known as CEP, is one of the 7. And it was established in 1981 in Montego Bay, Jamaica, to protect the coastal and marine ecosystems of our region, the wider Caribbean region. So I will give you some information on the Cartagena Convention. It was signed in 1983 under the auspices of the Caribbean Environment Program, as I mentioned before, and has remained the only legally binding agreement in the wider Caribbean region. The Cartagena Convention Secretariat, as Gillian mentioned, is based in Kingston, Jamaica, and is responsible for implementing the convention and its 3 protocols. The protocol concerning cooperation in combating oil spills that was also signed in 1983 and entered into force in 1986. The protocol concerning specially protected areas and wildlife, the SPO protocol, that was adopted in 1990 and entered into force in 2000, and the protocol concerning pollution from land-based sources, and also known as the LBS protocol. This was the last one adopted in 1991 and entered into force in 2010. So the work of each of the 3 protocols is supported by regional activity centers. For example, the oil spill protocol is supported by RAC Rainprotect Caribe, who is based in Curaçao. Our SPO protocol is supported by the SPO Regional Activity Center, who is funded by the government of France and is based in Guadeloupe. And the LBS protocol has 2 regional activity centers. One, the Institute of Marine Affairs is based in Trinidad, and the CMAP is based in Havana, Cuba. So as this presentation is, this webinar is related to plants, I was invited because I coordinate the SPO program, the SPO protocol. And here we have the main objectives of the SPO program, which are based in the main articles of the SPO protocol. And these objectives guide the work of the Cartagena Convention Secretariat and the SPO Regional Activity Center to implement the SPO protocol. So the first one is increase the number and improve the management of protected areas in the wider Caribbean region among the contracting parties. The second one is support the conservation of threatened and endangered species and the sustainable use of natural resources. Also develop strong regional capability for coordination of information exchange, training, technical assistance between the contracting parties, countries, and partners, and coordinate activities with relevant international biodiversity treaty and initiatives. Some of them were already mentioned by Gillian. For example, the Convention on Biological Diversity, and now with a lot of emphasis in support the implementation of the Kunming-Montreal Global Biodiversity Framework. Also, we foster collaboration with the Ramsar Convention on wetlands of international importance, the Convention for International Trade, and in endangered species of wild fauna and flora, CITES. I wanted to highlight that CITES has over 33,000 plant species, and some of these are also included in the SPO annexes that I will comment before. Also, we promote collaboration with other regional CIS programs and several initiative projects and regional organizations with similar goals to the SPO protocol. And on this map, you can see the countries that have ratified the SPO protocol today. There are 28 countries in the region, including these countries that have several territories like France, the United States, the United Kingdom, and the Netherlands. So of the 28 countries in the region, 26 have ratified the Cartagena Convention and the SPO protocol, the Cartagena Convention and the Oil Spill Protocol, and 18 have ratified the specially protected areas. So this one, the specially protected areas and wildlife protocol. So these 18 that you can see here. And one of our critical tasks in the region, one of our main works is raising awareness among non-contracting parties to supporting and guiding them through the ratification process. So our goal is of course, to bring all the 28 countries in the region to be part of the Cartagena Convention and all these three protocols. So how does the SPO protocol work? All the actions developed in support of the SPO protocol are implemented within the framework of biannual work plans. And these work plans are prepared in collaboration with the contracting parties, and then these work plans are reviewed by the Scientific and Technical Advisory Committee that meet every 2 years, and then these plans are endorsed by the SPO Conference of Parties, and finally these work plans are approved by the Intergovernmental Meeting and the Cartagena Convention COP. And the implementation of these work plans is guided by a working group of experts, also implemented through projects and also in collaboration with our regional activity networks and MPA networks in the region. So considering the topic of this webinar, I would like to highlight the 2 articles of the SPO protocol. The Article 10, directed to the establishment of national measures for the protection of wild flora and fauna. And in this article, parties are required to identify endangered or threatened species, establish measures to protect and recover these species, and identify, regulate, and prohibit activities that can affect this, that can have an adverse effect on such species. Also formulating policies and plans for the management of these populations. And then Article 11 that promotes cooperative measures for the protection of wild flora fauna, so collaboration between the countries. And in this, under this article, parties should establish cooperative measures with other parties to protect these species included in the annexes. The SPO protocol has 3 annexes that I will comment now. And also this article promote the establishment of a specific regulation for these species. So the countries need to protect these species in their national jurisdiction. And these are the 3 annexes. Annex 1 is the agreed list of species of marine and coastal flora that require full protection. So countries must protect all these species, included the plant species included in Annex 1. The same for Annex 2, but Annex 2 is the agreed list of marine and coastal fauna, so animals, to be fully protected. And then Annex 3 include species or of flora and fauna that may be used on a rational and sustainable basis, so require management measures. And quickly, as you can see in this graph, most species were added to the annexes in 1991, so almost 30 years ago. And some species have been included in the SPO annexes years later, no? But I want to highlight that only one plant species have been added to the annexes after 1991. So all the species included in the annexes, all of the plant species, except this one, were added in 1991. So with this, in particular with this graph, I want to highlight a key challenge for plant conservation, because the plant species included in the annexes were listed over 30 years ago. And another issue is that the conservation and restoration efforts for species included in the annexes have been predominantly focused on animal species. And also most projects addressing plant species included in the annexes have been focused on mangroves and to a lesser extent on seagrass beds. So to finalize, I think this challenge creates valuable opportunities for collaboration with plant experts who can significantly support the implementation of the SPO protocol. So, for example, one key opportunity is to nominate plant experts from the countries that are contracting parties to the SPO protocol, or from observer organizations. So we have a group of experts, and it would be great if plant experts can be part of this working group. And then plant experts can also provide technical support to the SPO contracting parties in crucial activities like designing and implementing plant projects and conservation and restoration initiatives. The plant specialists can also support the countries to add new plant species to the SPO annexes to ensure these annexes remain relevant and up to date. And also, of course, they can provide technical support to non-contracting parties to the Cartagena Convention and the SPO protocol to ratify. So these countries that are still not contracting parties of the SPO protocol can be guided through the ratification process with support of plant experts. So in conclusion, addressing all these challenges, I think presents significant opportunities for plant scientists to strengthen conservation efforts and to support the implementation of the SPO protocol. So thank you very much. I'm sorry to take a few more minutes.
Thank you very much, Dr. Susanna, for that really great overview of the SPO protocol. If you've got questions for Dr. Susanna, feel free to put them in the Q&A box, and we'll try to get to as many questions as we can at the end of the session today. Dr. Susanna, could you please stop sharing your screen and I will move forward. Thank you very much again. Okay. So I'll just quickly introduce myself. My name is Alicia Quinn, and I am a PhD student at Monash University in Australia, where I look at understanding the regulation of specialized metabolism in sorghum bicolor. But that is enough on me. I'm very, very pleased to be able to introduce Professor Casey Barnsall, who will be giving our next presentation. Professor Barnsall is a renowned expert in plant biotechnology and genomics, and he has made really significant contributions to developing climate resilient crops throughout his research career. He's focusing now on more policy and advocacy roles. And he has served as the director of ICAR's National Bureau of Plant Genetic Resources in India, and on numerous other boards involved in plant biotechnology regulation. So I will now pass over to Professor Bansal, and he can tell you about his work.
Thank you, Alicia, for the kind words of introduction. Greetings to you all from India, and thanks to the ASPB for giving me this opportunity to talk to you in this Plantae webinar on the topic Evolving Regulatory Framework for Genome Editing in Plants. And I'll give you an overview about this particular topic, and will like to cover briefly the historical perspective, that why biosafety regulation is really important around genetically modified organisms. You know, since they're students and young researchers, I thought it's important to talk about briefly the historical perspective. Then I will cover specifically the biosafety regulation around GM crops, and then followed by international agreements covering biosafety regulations of GM crops, genome editing and exemption of genome editing crops from biosafety regulation, and then current global status of regulation around genome edited crops in the world. I would like to just give you a brief overview on that as well. Just to start with the historical perspective, as you know, 1970 marked the emergence of a new discipline called molecular biology. And it all started with decoding the double helix structure of DNA in 1953, followed by the discovery of restriction enzymes in 1965, which made it possible to join one particular DNA, as shown here, the lambda DNA, with the DNA of the simian virus SV40 by Paul Berg in 1971. And this gave rise to the first recombinant DNA molecule. And with this, there was a new beginning of the laboratory applications of molecular biology from 1970 onwards. And the applications of recombinant DNA technology were realized. And to start with, as you know, that rDNA, also called a genetic engineering, allows the insertion of a fragment of DNA. DNA means containing one or more genes into another DNA, with the aim of modifying the genome of living organisms, and then also, to generate organisms with new properties through the manipulation of DNA. And this is how the first genetically modified organisms were developed. But then some pertinent questions were asked, for example, whether the combination of DNA sequences from entirely different species, even unrelated, will result in a new type of pathogenic organisms. That's the question which started bothering Paul Berg that time in 70s and other scientists. So in other words, do we need to therefore subject these GMOs to biosafety regulation? And since the SV40 DNA, which was used by Paul Berg, is an oncogenic DNA virus and can cause infections in humans, and is also found in monkeys, and is known to induce brain and bone cancers. So Paul Berg in 1972 wrote a letter along with 10 other researchers to the journal Science. And through this letter, he urged the National Institute of Health in US to regulate the use of this recombinant technology, and meanwhile also urged the scientists around him to halt this technology use or in other experiments until they understood safety around it, because this is the first time that Paul Berg discovered this kind of a recombinant technology, and they were quite worried about the regulation part of it and whether it can cause any kind of a harm, to human beings and to other organisms on this planet. So therefore, the safety issues of manipulating this DNA from different species were discussed in few conferences in Asilomar. For example, the Asilomar conference first was held in 1973, then again in 1975. And they concluded in the 75 conference that research work involving rDNA technology will be done following strict biosafety guidelines. So that was the great message which emerged out of these various discussions right from 1973, followed by even a Gordon conference, and then followed by the 1975 Ashlamore conference, that this research work involving recombinant technology will be done following strict biosafety guidelines. And NIH in 1976 developed these guidelines for research involving recombinant DNA molecule. That's how the biosafety regulation started of the GMOs. And these guidelines developed by NIH since 96 has been revised, a number of times and are currently in use. Now, the birth of this molecular biology in 1970 also gave us an opportunity to develop the first transgenic plant in 1983, followed by developing a series of GM crops around the world, to address the food and nutrition security. And currently, more than 200 million hectares are under, those GM crops in about 29 countries. To start with developing a GM crops, we need to develop the gene construct. Again, here we're using this technology, rDNA technology, and this suitable construct requires to have a promoter. For example, we have a promoter, mostly in plants from a CAMV, cauliflower mosaic virus 35S promoter, coding region from a bacteria, for example, BT gene, and terminator could be from another organism altogether. So this is again kind of recombinant DNA technology to develop a GMO, here in this case, a GM crop. And using these particular steps, for the development of GM crops, this GM particular crop is developed, wherein a BT gene, for example, is added. And then these GM crops are subjected to biosafety regulation. And why biosafety regulation? Again, because there's a use of rDNA technology. And this technology allows us to have a novel combination of these genetic elements. This is from virus, promoter, and this is from bacteria, and this could be still from another organism, and all unrelated to plants. And therefore, these GM crops are subjected to biosafety regulation. And for this biosafety regulation, international guidelines and national biosafety frameworks have been developed. These international guidelines were mostly developed based on the recommendations of OECD, the Organization of Economic Cooperation and Development, which was established in 1960 as a group of about 38 member countries. So OECD, in fact, published in 1986 its first safety considerations for genetically engineered organisms, including GM crops, which are relevant to human health, environment, and agriculture, and published as a blue book because the cover is blue. And as per this safety considerations, GMO development, or in particular, let's say GM crop development, are required to undergo stepwise risk or safety assessment test, right, starting from laboratory to growth chamber and greenhouse, then to limited field testing under confined conditions, and finally, the large-scale field trials are carried out before environmental release or commercialization of this to grow in the farmer's field when we talk of GM crops. And OECD gave a concept of case-by-case assessment of safety of these GM crops using certain guidelines and procedures, as I mentioned before. And also, these OECD recommendations became a major resource for the formulation of national regulatory framework around the world, and also the international regulations, including particularly the Cartagena Protocol on Biosafety, which came in force in 2003. The Cartagena Protocol on Biosafety is one of the international agreements regulating biosafety of GMOs around the world. And this particular Cartagena Protocol is a supplement, actually speaking, to the Convention on Biological Diversity. And biological diversity, I'm sure you all know, is an internationally legally binding instrument, which has these three very important objectives. One, conservation of biological diversity. Two, sustainable use of biological diversity. And three, fair and equitable sharing the benefits using plant genetic resources or germplasm in general. And to take care particularly of these two objectives, CBD required the contracting parties, those who are interested in developing GM crops or GMOs, to regulate, manage, or control the risks associated with the use and release of GMOs, or LMOs, you call them, resulting from biotechnology, which are likely to have adverse environmental impacts that could affect the conservation or the sustainable use of biological diversity. So therefore, this Cartagena Protocol on Biosafety, came into force in 2003 with the aim to ensure the safe handling, transport, and use of living modified organisms, LMOs, resulting from biotechnology that may impact biological diversity, also taking into account, importantly, the potential risks to human health. The next international agreement is International Protection International Plant Protection Convention, IPPC, which came in force in 1951. And the aim of this particular convention is to prevent the spread of pests affecting plants and plant products. So therefore, IPPC adopted GMO Act in 2004 and identified certain potential pest risks, of GMOs. For example, GMOs may cause invasiveness. So we need to, study about the invasiveness of the resultant GM crops. We're talking about it before they are environmentally released. Or the GMOs or GM crops could transfer, genes from GM crops to wild relatives or other compatible species via gene flow. Or GMOs could affect the non-target organisms, particularly beneficial insects or birds. And the third international agreement regulating biosafety of GM crops or GMOs in general to do with food safety. And here, this is the Codex, which came in force in 1963, the Codex Alimentarius Commission, which is an international food standards body established by FAO and WHO to protect consumer health and ensuring fair international food trade. So therefore, to ensure the safety of GM crops or GM food, in 2003, the CAC adopted principles of risk analysis of foods derived from GM crops. So in addition to the environmental protection, here food is something important. And therefore, under this particular, Codex Alimentarius Commission, the governments are desired to have test and approve the safety of GMO-derived food before it enters the market, because they are ensuring the fair international food market and consumer health. So therefore, before it enters the market, these are tested, and the GMO food should be tested for the potential to cause allergenic reactions or toxicity for that matter. And based on all these international regulations or regulatory or procedures and guidelines, national biosafety regulatory frameworks were developed by different nations, around the world. I'm just giving an example here in case of India, which I'm more familiar with, that we developed, for example, these are all revised over time. Latest we have is 2016, where all those safety assessment tests are done right from laboratory research to greenhouse to field testing before their environmental release and commercial cultivation. And what kind of tests and data are required for safety assessment of these GE, we call it in India, like genetically engineered or GM crops, they're the same. For food and feed for food and feed safety, toxicity assessment is done, assessment of allergenicity, etc., or composition analysis are all done. I'm not going to go into the details of it. And for the environmental safety, specifically, I said before, the impact on non-target and beneficial organisms like predators, soil microflora, etc., are tested. And also the changes in gene flow pattern, if any, through pollen flow studies is analyzed and studied, and particularly the crossability studies of that particular GM crop, which has been developed with sexually compatible relatives, which if they're grown in that vicinity, in that area, they're all studied before they're allowed, by these biosafety, regulatory framework of a particular nation, for example, here in the case of India. So that was all about GM crops. That was important as a background to really talk about the, biosafety regulation of genome editing. And genome editing, as you know, is a targeted mutagenesis procedure when it starts with creating a double-stranded break by the Cas nuclease. And this double-stranded break is then repaired, with three different mechanisms. One is this non-homologous end joining, where some certain randomly, some bases are inserted or deleted. Indels, we call them, causing the gene disruption. We call it as delete in terms of editing point of view, and it is referred to as SDN1. The second is that certain point mutations or certain bases, few of them are replaced or changed, using a homology-directed repair mechanism, wherein a template is provided, which is then added here and allows a gene correction. We call it as edit2 and referred to as SDN2 category of genome editing in plants. And the third category is, actually speaking, SDN3, which is like exactly equivalent to GMOs or GM crops, wherein a foreign gene, as I mentioned before, in case of GM crops, is inserted, except that it is inserted here through a process of homologous recombination into a well-defined, identified location on a genome. So that's the difference. When you develop GM crops, it gets integrated randomly. But here it's also a GM crop, but it is integrated on a predefined, site. So now, when you come, the question was asked whether genome edited, or genome editing in crops to be regulated like GMOs. And as also now, if you compare SDN1 and SDN2 with SDN3 or insertion of a foreign gene, here the small changes are made only in the existing genes, in the native genes of the host or of a given plant. And also these two categories, SDN1 and SDN2, do not result in any novel combination. We don't bring in any gene or a promoter from a virus or from bacteria. So there's no novel combination of genetic elements. And as such, these genetic changes, particularly in SDN1 and SDN2, can also be achieved through could be achieved naturally or through conventional breeding. That is also has been analyzed and mentioned about it. And therefore, there are crucial differences, really speaking, when you compare genome editing with genetic engineering. In case of genetic engineering, I mentioned before, there is a precisely insertion of a foreign gene, giving rise to a GM crop. But here, in the case of gene editing or genome editing, natural small changes only take place, and that too within the existing genes, and there's no foreign gene. And I said before that in SDN1 and SDN2, such genetic changes could occur even through conventional breeding. So therefore, if you now try to answer this question, scientifically, we would say that these genome editing crops are not really particularly SDN1 and SDN2 to be treated as GMOs or to be regulated as GMOs. And since we have seen that very precisely, kind of changes which are made, particularly in SDN1 and SDN2. So therefore, several countries have exactly exempted SDN1 and SDN2 crops, from biosafety regulations, and based on following criteria. I mean, different countries have mentioned it or worded it differently. For example, some of them, they say that there should not be any new genetic material introduced as in SDN1. So they will not treat these crops as GMOs. Another country says that there could be an absence of an externally provided repair template or no foreign DNA present in the final product. Absence of transgene is again kind of foreign DNA in the final product. Absence of any exogenously introduced, again like foreign DNA or transgene in the final product. No novel combination of genetic material to be involved. Have genetic changes that could also occur naturally or could be achieved through traditional breeding. And particularly SDN1, again, just as some other countries mentioned, this is overall, I'm taking from some examples, and cisgenesis, where there's introduction of a gene present in the plant gene pool or the varieties or could be even compatibly sexually hybridized to transfer that particular gene. So such genes or even single base pair substitution, they are supposed to be all not treated as GMOs, and they fall under the category of either SDN1 or SDN2. So keeping all this in view, if you look into the regulatory status now, let's say as of July 2022, several of these countries, in fact, there are 18 of them as shown in green color, like Canada, US, and many of these countries shown in green, they have taken genome edited crops without the foreign DNA, particularly again SDN1 and SDN2, to be not regulated as GMOs, as shown in green. But there are certain countries, like 4 of them, as shown here, Norway, Indonesia, Bangladesh, Uruguay, where the proposals are under consideration, or let's say were under consideration. I'm talking in July 2022. However, European Union or New Zealand, they still consider genome-edited crops to be regulated as GMOs. And actually speaking, USA taking a lead forward, they granted exemption, 63 exemptions covering 17 crop plant species as shown here, 17 of them, soybean, the maximum about 20 out of 63 for various traits. And we in India as well made enough efforts, really speaking, I was personally involved to persuade the government of India to exempt genome editing in plants, from biosafety regulation. During this particular period, we were making all those efforts from 20 to 2022. And we finally succeeded. And India announced exemption of genome editing in plants from biosafety regulation, in 2022. And we developed these standard operating procedure SOPs in October 2022. And currently, in fact, 2 different crops, so 2 rice events developed with DST gene edited, which has got 25% higher water use efficiency and about 10 to 20% higher grain yield. And the other one with, more cytokinin, content in the meristematic tissue with higher productivity, they are undergoing field trials as conventional bred crops. Because in our SOPs, we have declared, that if the absence of foreign DNA is scientifically proven, they are to be treated as conventionally bred crops. So these two have been tested and passed through that procedure using these standard operating procedures. I'm just giving an example from India, and they are undergoing field trials. 2023 was done last year, and they are going in this particular season right now in the field in this year, 2024. And more are in the pipeline in the production system. So in the meanwhile, globally, regulation of gene editing technologies in crop is making progress. Now let's compare today, now 2024, with the previous data I showed you on 2022. Now the exemption granted by several countries, the number has increased from 18 in 2022 to 25 in early 2024, Thailand being the 25th country. And again, those countries where there's active consideration going on the proposal on genome editing, 4 in 2022, now the number has increased to 12 in 2024. So what's going to be the result? We have to wait and watch. But in the meanwhile, there are certain countries, as shown in purple here, like European Union, many other countries, and also New Zealand, they are still treating genome editing in plants as GMOs. And but then there are certain good news coming across, as you can see actually in July and August, European Union, for that matter, a legal proposal is already, being negotiated, on new genomic technologies that includes both genome editing and targeted insertions of cis genes. That we hope that the result will come out soon. And also in the meanwhile, there's a declaration by the agriculture minister in the New Zealand that genetic engineering ban to end by 2025. So there's the positive news coming across, and more countries are joining, and declaring SDN1 and SDN2 particularly, even cisgenesis, which could be a part of SDN3, targeted insertion of these cis genes, as SDN3 will be not treated as GMOs. So I would like to conclude that genetic changes in the existing genes, as we have seen in case of SDN1 and SDN2, not regulated as GMOs in several countries. We have seen that, including targeted insertion of cis genes. However, regulation of gene-edited crops is still evolving, and currently there are no international consensus emerging out of it. So therefore, what is needed is globally, or even regionally for that matter, I would prefer globally overall. I'm sure we all will prefer, because we need to have a free trade, and these guidelines should be all similar around the world, to be harmonized, have a harmonized regulatory framework or guidelines to harness the full potential of genome editing in crops for global food security and sustainable agriculture, which is so very important when we talk of climate change. So with this, friends, I thank you profusely for listening to me patiently, and I hope I have not overstepped the time, and I'll be happy to answer if there are certain questions. Thank you again to the organizers.
Thank you so much, Professor Bansal, for taking out time to speak on this important topic and for such an important and informative talk of yours. Alicia, if you may just go to the next slide.
Is it okay now? Thank you.
Hello, everyone. I'm Indrani. So I'm a Rice Postdoctoral Fellow and an ASPB Plantae Fellow. So I'm here to moderate this part of the webinar. It's an important topic. So without spending much time on introductions, I would just like to say a special thanks to one of our other Plantae Fellows who couldn't be here due to her work emergency. I would like to introduce our next speaker, Dr. Felix Moronta Barrios. He is a senior program assistant with the International Center for Genetic Engineering and Biotechnology. He's a Venezuelan biologist committed to facilitating the adoption of agricultural products enhanced by modern biotechnologies. As a senior program specialist in the regulatory science group at the ICGB Italy, he supports projects to energize and strengthen biotechnology regulatory offices in the Global South. He works closely with national and international authorities for ensuring regulatory effectiveness in the changing landscape of evolving biotechnologies. With this, I would ask Dr. Moronta to please give his talk. Thank you.
Yeah, thank you so much, Indrani, for that very kind introduction. I'm going to proceed to share my screen. Okay, and I wanted to start just by telling you that I am here in these campuses. I am talking to you from the city of Trieste in Italy. We are in a very nice corner in this country. And in one of these buildings, is my institute, the International Center for Genetic Engineering and Biotechnology. And today, I wanted to talk quickly about regulatory science and how it is applied in emerging agricultural biotechnologies. As Indrani said, I am a senior program assistant in the regulatory science groups. Here in the International Center for Genetic Engineering and Biotechnology, I repeat, we are an intergovernmental organization with more than 60 member states, mainly from the Global South. And we have components also in Cape Town, South Africa, and in New Delhi, India. And I'm going to discuss also some activities of my group in this area. So to begin, let's start. Let's take a closer look to the foundational concepts of regulatory science. General definition that is provided by the FDA. Regulatory science is a scientific discipline consisting in the development and application of scientific methods to support regulatory and policy objectives. And it is focused on the scientific basis and also concerns of regulations rather than promulgating, implementing, complying, or enforcing regulations. On the other hand, as Professor Bansal already introduced, biosafety, just as a summary, is all measures, policies, and procedures necessary to minimize the potential risks to the environment and human health resulting from modern biotechnology. And this is a short explanation that is that was formulated by the Convention on Biological Diversity. So we can differentiate regulatory science because establishes the frameworks and methodologies needed to evaluate biotech products. Instead, biosafety ensures that these products are assessed and managed in a way that protects environmental and human health. And now that we more or less know the importance and difference between both, let's explore some of the cutting-edge biotechnologies that are emerging in the agricultural sector. First, it was already perfectly introduced by Professor Bansal, is about genome editing. So I will jump straight to the next one, and it is about gene drives. So this is another emerging biotechnology that I wanted to highlight today. And the advances in synthetic biology, for instance, including CRISPR, have delivered molecular tools that are necessary and computational tools too that are necessary to design and develop a wide range of engineered gene drives. And these elements spread genetic modification at rates that are greater than expected from a normal or Mendelian inheritance. And these characteristics of gene drives enables them to proliferate rapidly within sexually reproducing organisms. And consequently, this approach of engineered gene drive holds considerable promises in tackling challenges concerning, for instance, the management of harmful populations. And unlike others or classical GMOs, the engineered gene drive are often designed to be applied in wild organisms. And also they are designed to spread beyond their initial release location and persist in the target population over time. And there are, quickly, there are basically two strategies. One of them is the population control using gene drives. And the other one is population suppression. If you can compare here the introduction of transgenic, in this case, mosquitoes, in this case, the introduction of the gene drive mosquito could replace the population of wild type mosquitoes. In this one is another approach. The introduction of the gene drive mosquito will provoke the collapse of the total population of mosquitoes. And as a result, as you can imagine, the ecological impacts of the deployment of this kind of organism could be far reaching, of course. And while gene drives are well developed in yeast and some animal, like mosquitoes, and perhaps also in rodents, in mice, applying this technology in plant has been a major technical challenge. However, this is changing. Now genetic intervention in wild plant population is technically possible, or according to the latest research and models. For instance, recently, in June, a couple of months ago, two groundbreaking studies, the one that you are seeing there, were published simultaneously that successfully introduced gene drive elements in Arabidopsis. And these two teams independently engineered Arabidopsis to carry a trait that is inherited by the 99% of the offspring. And they calculated that within 10 to 30 generations, the entire plant population could harbor the genetic trait that they introduce. So this kind of research and finding bring us closer to the potential application of gene drive in agricultural fields. And given these advancements that are happening now, one might ask, how is the regulatory landscape evolving to accommodate such transformative technologies? We are talking about releasing just few genetically modified individuals, and the genetic modification will spread with the field. So this is a current landscape on gene drive regulation. There are, only 2 countries that has specific mentions, at least, to gene drive technologies in general. But most of the country are strongly regulating the technology. Most of the countries that you can see in this list, because most of the countries of the world are not even mentioning the gene drive in their regulatory frameworks. So the engineered gene drive present unique regulatory challenges as new emerging technology, biotechnology in this case, and the potential to bring profound change in the ecosystem and species dynamics because of this. And there is the need of a comprehensive regulatory framework to ensure their safe and responsible use. And well, in my group, we are well positioned to support the development of science-based regulation, to provide scientific expertise in the creation of regulatory frameworks, and also not only to create, but also to update these regulatory frameworks, and helping to operationalize existing regulations, and all of this to ensure the safe use of biotech products. And one key aspect of all this process is the implementation of solid and sound environmental risk assessment. So I want to talk about just one slide about this aspect, and the environmental risk assessment of gene drive. So environmental risk assessment is a cornerstone of biosafety, helping regulators understand the potential impact of new plant varieties and ecosystem. Risk assessment is a process based on available scientific evidence that is aimed at informing the decision-making process, and the methodologies and outcomes may differ between countries and jurisdictions. And this is important, it is always emphasized the relevance of performing a case-by-case risk assessment, because there are two genetically modified organisms are not the same. No, it depends on many variables. And the environmental risk assessment is focused on, well, basically in trying to answer with scientific evidence for these four questions. What could go wrong? Please, the audience, if you can imagine a potential gene drive plant or a genome edited plant, please try to respond in your minds. No? What could go wrong? How likely is the harm to occur? How serious could the harm be? And what is the level of concern? So, and properly addressing and documenting this critical question is crucial for assisting decision-making in determining whether a new technology can be safely deployed or not. As we have seen, ensuring the safety of biotechnology requires not just sound science, but also robust and evolving global policies. And talking about these policies, I wanted to bring a concrete example that my group was involved recently. And collaborating with the Convention on Biological Diversity, we have developed guidance for the risk assessment of genetically modified organisms containing gene drives. First, to know what is discussed in the scientific literature, we perform a literature review. I'm not going into the detail here, but I will be happy to elaborate more separately by email or whatever. But the thing is that we gather all the scientific literature talking about risk assessment of gene drive organisms. And with this approach, we could confectionate, let's say, this guidance that consists in all these points that you can read here. I'm not going to go through all the points because the lack of time, but this is something that it is already published, is publicly available. And this risk assessment guideline will help operationalize national regulation to ensure that the engineered gene drive can be used and deployed while safeguarding biodiversity. These guidelines were more focused on mosquitoes, but again, it could be tailored to agricultural products too. And other example of our activities here in the ICGEB, in the area of international policymaking, I wanted to highlight that we were the author of this comprehensive report that we developed for the Convention on Biological Diversity on synthetic biology. It was published a couple of years ago, and has been used and is being used by the CBD party for the discussion on the topic. And now we are working closely with the International Union for the Conservation of Nature. This international organization is developing their institutional policies on synthetic biology in relation with nature conservation. There are many applications that could be used for the benefit of biodiversity, so they are starting to discuss in that topic, and we are helping them in those discussions. Regarding regional and national regulation, we have worked closely with the UN Environment Program to provide technical assistance to Caribbean countries that you can see there, and also to the government of Panama to develop for these small states and to update, in the case of Panama, the regulatory biosafety framework. And also we provide capacity building activities and capacity strengthening activities national or regional approach, basically in risk assessment of genetically modified organisms. And now we are tailoring our training to be more focused on emerging biotechnologies of genome editing and gene-drive organism. With this, I conclude, and I'm interested for you to take these messages with you, is that regulatory design is essential to ensure that agricultural biotechnologies are safe, effective, and with benefits. Technologies like genome editing and gene drive offer transformative potential for agriculture, and at the same time, unique regulatory challenges. The agricultural biotechnologies are constantly evolving and faster than regulations. So it is needed a science-based regulatory frameworks that can keep pace with technological advancement. Again, regarding my group, is that through technical assistance, capacity building, and international collaboration, and this is and international collaboration, we facilitate or help to facilitate the safe deployment of agricultural biotech. And the final message is that policymakers, regulator, and international organization must work together to create an adaptive, future-proof regulation that support the safe and innovative use of emerging biotechnologies in agriculture. And with this message, I rescued one of the conclusions of the Professor Bansal, asking for harmonizing regulations around the world. So thank you so much for your attention. I will be super happy to respond some questions that may arise.
Thank you so much, Dr. Maronta, for your talk. We indeed know a lot now after the end of this webinar. We are currently in our Q&A session. Without spending much time, I would like to forward the questions. A few of them have been already answered by Dr. Susanna. And I think I'll straight away go to asking it then to the speakers. The first question, I think we should go to Professor K.C. Bansal. The question goes, what are the current measures to protect the open pollinated varieties, especially the many maize land races in middle and low-income countries, low-income economies, where BT and other transgenic maize are both allowed or likely to be grown?
Now, that's a good question. You know, each country, like we have in India, a facility to, to conserve, to collect and conserve all those land races or open pollinated varieties, even farmers' varieties, and they're all conserved in these national gene banks. And I'm sure the country we come from, they would certainly have that as well. And over and above, there is one international gene bank, which is supported by Crop Trust, Global Crop Diversity Trust, which is based in Germany. They have an international gene bank where the countries are also, allowed and encouraged to keep their germplasm safe. So what I'm trying to say is, irrespective whether these genetically modified organisms and crops are spread, which are of course required for the benefit of the country as a whole, or for the consumers, for the quality product availability point of view, but at the same time, there's a very conscious effort on internationally to conserve all these land races, including, I mentioned before, farmers' varieties or open pollinated varieties. Thank you.
Thank you so much for your answer. Yeah. Moving on to the next question. This is, I guess, aimed at Dr. Maronta. Why gene drives are challenging in case of plants? And what is the difference between the evolving regulatory, I don't know, I can't get what ERA means though. I think evolving regulatory association of GMOs and gene drives.
Okay, well, for the first question, it was about why gene drive are challenging in plants, no? Isn't it?
Yeah. Right.
Okay, well, shortly, plants have a much bigger, complex reproductive biology than insects, or that mosquitoes, Anopheles mosquitoes or Aedes mosquitoes, and more complex than mice. So the also plants has a slower breeding cycles, and plants has a potential of polyploidy. So this kind of intrinsic characteristic are putting difficulties in achieving efficient gene drive, the gene drive inheritance. No? Remember that each of the seeds and each of the pollen should then harbor, each of them, the genetic modification. So because of the biology of the plant and the animal, it has been developed faster in animal this time. One of the difficulties, in the other way around, plants has the advantage that yes, gene drive can modify a wild plant, but the plants are not moving around like a mosquito or a mice. So this is a compromise between these two approaches. And okay, and the second question was more related to environmental risk assessment. Let me read it again.
Yeah, it's differences between the evolving regulatory, I think, rules of GMOs and gene drives.
Okay. Okay, well, the environmental risk assessment of GMOs, we have a lot of experience on that, decades of experience in environmental risk assessment of GMO, and they are mainly focused in assessing the impacts on non-target species, let's say, non-target species and the environment. Instead, the environmental risk assessment for gene-drive organisms also consider the potential for a rapid and uncontrolled wide spread of the genetic changes in wild population, also potential irreversible ecological impacts, and well, the challenges of containment, or the management of potential risk once they are released. And because of this, huge challenges in gene drive organism. One of the characteristics of this technology is that it would be extremely difficult to, or perhaps impossible, I don't know how to interpret it, to perform field trials, no? Because once you release one of these organisms, they could potentially spread. So the challenges are now how to contain, no? How to get data, first of all. So you need a field trial to get data, real life data, but with a gene drive organism, it's extremely difficult. So now the research is, the research is contained in laboratory settings, and the discussions and many proposals are now on how to eventually, evolve and try to get data from the real world.
Well, thank you for your answer, Professor Dr. Moranto. Yeah, going to the next question, direct this to Professor Bansal. What are the problems associated with genetically engineered plants, and what are the benefits of GMOs and GEDs in climate change?
Well, both the technology is, GMOs or GM crops, genetic engineering, and genome editing are important, and both they have a very important role to play, while developing climate resilient varieties. But the only advantage with genome editing, which we have seen over the last about a decade, is that they are not regulated as heavily. And the process is quite eased out with regard to biosafety regulation. So there's a lot of saving of time and resources, and the process is quite efficient and targeted and effective in developing any crop variety for that matter for climate resilience. But however, in case you need to precisely transfer a foreign gene for a particular trait, GM crop technology still is the choice. People are talking about it. And though you can have the equivalent advantage of overexpression, now we have got certain, mechanisms wherein Cas CRISPR-A activator, wherein you can regulate the expression, enhance or suppress through genome editing. But people still prefer at times tissue specificity point of view, those promoters, which can be used through the process of developing GM crops. So both are important, of course. Now, since we have seen the advantage of reducing stomal density or increasing water use efficiency, even the examples of reducing methane gas emission, for example, through GM technology. So I would say both, they have their own advantages. I would say to decide upon which crop, what trait you want to do it, but certainly with regard to efficiency, with regard to effectivity and the ease out process with regard to regulation, genome editing will always remain, kind of technology of choice. And more, I'm sure if you can email me, I would be very happy to take up, since because of the time, maybe there's another question, I would like to take up that if there's any. Thank you. Yeah, for this question, please. Indrani, over to you.
Indrani, you're muted.
I'm so sorry. There's another question directed to you, Professor Bansal. If you could quickly, very briefly answer that.
Yes.
Could you elaborate the IIRR variety, which you mentioned in your talk?
Oh, yes, this is the variety wherein a particular gene, CKX2, which is responsible, which has an activity, enzymatic activity, the gene encoding in enzymatic activity, cytokinin, oxidase reductase, which will increase actually the level of cytokinins. And the cytokinin accumulation, particularly in meristematic tissue, plays a role in development and increasing the number of tillers. So therefore, if you mutate this gene, which was earlier shown by some of the Chinese scientists, through otherwise, kind of a molecular breeding approach, functional analysis of this particular gene, they found it's quite effective in increasing the yield. So here, that particular gene through genome editing has been mutated, and so that there's more cytokinin availability or functionality available in meristematic, increasing the number of tillers. So therefore, he has been able to achieve this higher number of tillers and more yield. And even the plants are early in flowering and robust culm, so there's certain bonus advantages as well. And we can discuss more about it again later. So this is what in brief. Thank you.
Thank you so much, Professor Bansal. I think there's two last questions. This is the second last. This is a general question. Whoever of you, it's open for any one of you. So do you see a future where sharing of germplasm between nations will become easier?
Can I take it?
Yeah, sure.
Actually, the germplasm sharing has always been easier since the introduction or the CBD in 1993. The process has no doubt slowed down. And because the process got slowed down from 1993 onward, 10 years later, we have this International Treaty on Plant Genetic Resources, Food and Agriculture, which came in to facilitate the exchange of germplasm through a multilateral system, through a standard material transfer agreement system. Because these germplasms, if they are used without any benefit sharing advantage to those countries, then certainly, they will not like to exchange it. So benefit sharing is something which need to be, kind of eased out, and Nagoya Protocol, and including even international treaty, has taken care of it very well. And there are international laws. If we follow those laws, particularly international treaty, there is no problem at all for exchange of germplasm. One can go for it, including from India and many other countries, those who are signatory, of this particular treaty and also the CBD convention. Thank you
so much. Thank you for your answer. One last question, and again, it's open to any of the speakers to answer. What is the main player at the moment for countries to have less attention on the potential benefits of GMOs and gene-edited crops and even EGD in Africa? So I didn't quite understand.
I will need some further explanation of the question, please.
Yeah, I think, a little
No,
confusing.
the question is not very clear. I think the person should himself clarify, or herself, yeah.
Yeah, I think we'll skip it then and would like to, they can ask it now too if they're still listening to the webinar, and we can directly put it to you. I think, Jason, can we take any more questions, or are we done for today?
I think we could do like that. I think there's only one last question, and then we'll wrap up.
So again, it's open for everyone, anyone who wishes to answer this. So what regulatory policies exist for plant breeders using nuclear technologies? Any
Using?
nuclear technologies?
Well, we have nuclear technologies like, introducing a new gene, into a nuclear genome.
Right. Right.
Or manipulating or editing or deleting that gene again from the nuclear genome. We use genome editing. But in addition to nuclear genome, we have another genome, actually speaking, in plants, that is chloroplast genome. So in fact, the chloroplast genetic engineering is also an own advantage, and which has been done in many cases. So there is a diversity there as well, and people are talking about it increasing more of either nuclear transformation or chloroplast transformation for crop improvement through genetic modification technology or through genome editing.
Oh, that's interesting, because I was thinking more in nuclear from radiation.
Correct, Felix.
Oh, I see. Of
Yeah,
course.
exactly. No, but it's extremely relevant, your intervention, Professor.
No, thank you. Nuclear radiation, yeah.
Yeah, nuclear radiation that has been used from decades ago, from
Yes,
I don't know, 70 years ago, in the 30s, something like that.
Yes,
So
yes, you're right.
this
For
is
mutagenesis.
yeah, exactly, long-standing and very extremely successful technique to increase the genetic diversity in plants. Actually,
You're right.
yeah, most of the, at least here in Europe, in Italy, the wheat that we consume in the pasta, in bread, in pizza, no, those wheat were developed using radiation and so on. And this is something that it has been extremely important for.
Yeah, there are more than 3,000 varieties which are available globally through irradiation or through chemical mutagenesis. You're right.
Yeah.
So to interrupt, so do we have any existing regulatory policies on that? So that was the question, basically. Do we have any existing regulatory policies on such?
No, there is no regulatory policy at all to control whether through chemical mutagenesis or through irradiation, any kind of, or conventional breeding for that matter, which it all includes the same. Only thing is the process are cumbersome, lengthy at times, takes a lot of resources for phenotyping and selecting the best. So therefore, we have this advantage of now having GM technology or further genome editing technologies.
Yeah, that's correct. They are treated exactly as a conventional breeding product and registered like that. No regulations.
Right. Thank you so much. Thank you to all the speakers for this wonderful webinar. And I'm pretty sure all the audiences have really gone back wiser in terms of, it's a big topic, it's a vast topic, and an hour is actually not enough to talk on all the policies, but thank you so much for taking out your time, and especially Professor Bansal. I know it's almost 10 o'clock in India right now. If you're
that's fine.
Yeah. And yeah, with that, I conclude this webinar, and over to Jason.
Thank you, Indrani. Again, thank you. Just want to echo the thank you that Indrani shared. And then also just a quick brief reminder again, we shared this in the beginning of the slide. If you want to be more involved in various world policies in plant science, again, AFPB has a really active science policy committee. So I highly encourage you getting more involved, reaching out to our community chair to be more involved in this topic. It's a very important topic and one that we should all kind of really be engaged with. Without further ado, I want to again thank you so much, and not to keep you any longer. I hope that you found this webinar really relevant and helpful, and we hope to see you on the next one. Thanks, everyone, and have a wonderful weekend. Bye, everyone.
Thank you, Jason. Thank you, everyone. Have a good
Thank
weekend.
you.
Bye-bye. Thank you so much.
Thank you
Bye-bye.
all.
Thank you. Bye.